Hello, everyone. Thank you for staying to the end. I have a brief presentation today. So I'm a group leader at King's College London, and I'm also a Welcome Career Development Fellow. My group is computational, and we develop methods for understanding tumor heterogeneity and identifying biomarkers from large-scale imaging data. So today I will present a method for classifying histopathological images. And the reason my group is interested in developing methods for these images is that they remain the gold standard in cancer diagnosis. So these images usually is basically tumor sections stained with the hemotoxylin and iosine, and these examined by the pathologist to look for changes in the tissue architecture and morphology. So if I zoom in one of these regions, you can see, just putting the pointer, you can see this small region corresponds to the hundreds of cells where each purple dot here is a nuclei, and the pathologist will try to identify changes in the morphology and how the nuclei organized, as well as if there is mitosis, increased mitosis, etc., to diagnose the patients and sometimes even get a picture of potential prognosis and how much the patient will live. So these images are very rich in information. However, we know as human vision can be subject to error. It can be variable depending on their experience. And of course, it can be inaccurate sometimes. So what we are interested in to do is to automate this process by using deep learning approaches and developing robust methods that can provide support systems for the pathologist in their clinical workflow. And the aim for that is first to increase accuracy, but also save time. So we know over the world there is a shortage in number of pathologists. We know that some pathologists will sit on the case for weeks, not being able to give diagnosis, which means that not only we increasing the treatment cost, but also maybe making things worse for the patient. So really having more automated systems can be valuable to address that. And of course, for my lab, the most important thing we are interested in is if we can discover new signatures and biomarkers for cancer patient treatment. So our aim is to come from image to predict the diagnosis and response without annotation. So just from the large image predicts the final label, and this fall under the weakly supervised learning because the region that we will associate with the diagnosis can be small part of the image. So it is like we have a subset of regions, and we're trying to look at any abnormal region. So to address that, what we do is to divide the image in small patches, and we filter the ones that are empty. From that, we process each patch through a pre-trained neural network to obtain features, and these are passed through a weakly supervised model. So there is a few approaches that have been developed, and the previous talk presented one of them, but today I'm focusing on two. So there is vision transformers, which is the same technology that is used by ChatGPT. So transformers has been shown to be very effective when we have a large amount of data. And instead of sequence of fours, vision transformers take a sequence of patches. So these very small patches are passed through a transformer encoder, which basically include multi-head attention mechanisms. So this mechanism basically tries to identify which patches should receive more weight, and based on that, it's passed to the next layer. And this is then followed by multi-linear projection unit that predict the final class. So this method is very powerful, but also very expensive because we're trying to process many images in parallel to also understand the dependency between them. Another approach called dual stream multiple instance learning, DSMIL, and these methods have two streams, one for classifying the instance, so the small patch or subregion of the image, and the bag classifier. And the bag here is the patches in the entire images. In the entire image from the patient. So how this works is basically the method will try to give weight for the instance here through a convolutional layer, and pass this information to the bag classifier that combine this information together to predict the bag label at the end. So this model is very lightweight, does not have many variables because there aren't many convolutional layers. So we wanted to investigate if we can make these models can compare similarly to vision transformers. So what my research assistant Danu did, he proposed a model that just increased the depth in the instance classifier by adding 2 linear layers followed by dropout and ReLU. And then this is the model, the rest of the model remained the same. So we tested these different methods on a glioma dataset from TCGA, and these have 147 images, 3 subtypes: anaplastic astrocytoma, glioblastoma, and oligodendroglioma. So it's quite balanced, nice data set that have a small data size, but also reflecting of data sets available currently. And what we found that actually DSMIL plus can outperform vision transformers called here VIT. We also tested a more advanced model of transformers, graph VIT, which consider the spatial relationships, and DSMIL plus, which we proposed, have comparable performance. But if you look at the time and the complexity of the model, DSMIL plus is much simpler, which means that it can be applied much more efficiently. One thing that was interesting that when we looked at the various model performance, we see that actually this can vary according to the subtype, which suggests, like, for example, for the astrocytoma, we see a graph VIT outperform the others, but for glioblastoma, the DSMIL plus outperformed others. And this suggests that these different models can be better at discovering some signatures versus other signatures, and maybe there is advantage in combining them. So in summary, DSMIL plus is a lightweight model for classification of histopathology images, and we think that is a good way to prototype, or when we have limited data, to use these models and maybe combine them with more complex models. So I think many people were recruiting today. We are also recruiting. We have interesting projects around analysis of multiplexed imaging data, as well as spatial transcriptomics. So if you are interested, please get in touch. We are based in Waterloo campus in London, so it's a quite nice place. And I would like to acknowledge Danu, who performed this work, and my funders, MRC and Wellcome Trust. And thank you for listening.
Thank you very much, Heba, for the nice talk. So we have a couple of questions, actually. I think we can take all 3 of them in that week. If not, I invite you to talk to Heba yourself.
Hi, thank you. Thank you for the great talk. So in the beginning, you said that you segmented the images, the histopathological images. Did you use any segmentation algorithms, or was it just based on the graph that they were on?
We didn't segment them. We segment the entire tissue just to filter the empty ones, but we did not segment the cells. So we just divide them into patches, and these patches are passed through a neural network to extract features. So it's quite low, like in human intervention, from that side, because segmentation can introduce errors, and we try to avoid those by using this approach.
Hi, thanks. So in the supervised setting, what are you learning, a single variable, or it's like a number of variables that you're learning?
In this example, we have the 3 labels. Yes. So.
Okay, yeah. And usually, what are they? Is it like?
So it was the labels, like in our case, just as different subtypes of glioma. So glioblastoma, astrocytoma, and oligodendroglioma. So it's like if you have more classes, that's fine. It's work like it can be extended.
Last question.
Thank you. It's really impressive that it works so well. So I'm wondering if you looked into the interpretability and learning how your model is making its prediction, and especially to go further, it would be interesting to see if pathologists can learn from the model, and the other way around, if the model could learn from the pathologist by making use of what is already known, like which features are already known to distinguish these different variants.
Thank you. That's a very important question, and it's actually the focus of my lab. So we started looking at the explainability, but actually we would want to also develop the model side by side to improve the explainability. So we can see some distinctive features from these images, but also we want to work with the pathologist now to ensure that what we're discovering sometimes can be artifacts and sometimes can be associated with the biology of the cancer. So it's like how we can ensure that the models discover and identify the right signatures.
Thank you very much.